Okay, let's talk about DNA, one of my favorite things to talk about. And as you guys know, DNA stands for deoxyribonucleic acid. It is what we talk about when we talk about genetics, and I just love talking about DNA. So in the case of when we talk about anything having to do with DNA, remember the number 3. There's 3 letters in DNA. There's going to be 3 parts to the structure that makes DNA. There's going to be 3 steps in making DNA. There's going to be 3 kinds of RNA. There's going to be 3 steps in the making of RNA. There's 3 steps in the making of a protein. Every 3 letters is going to mean something. So anytime you think about DNA or the whole process of protein synthesis, remember the number 3. Now, like we said, in the case of DNA, I always call DNA a cookbook because everybody knows what a cookbook is. Watson and Crick, the 2 gentlemen who came up with the structure of DNA, calls it a twisted ladder. Well, you don't read a ladder. You read recipes. You read books. And so whenever I think of DNA as a cookbook because we've all had cookbooks. We've all seen a cookbook. We've all seen recipes. We know what recipes are. Well, when we talk about genetics, that's what we're talking about. DNA is a cookbook. It has covers and it has pages. And the old saying is don't judge a book by its cover, because the cover isn't the important part. The written word on the inside on those pages is the important part. And that's how DNA is kind of structured. DNA has 2 sides that I always think of as the front cover and the back cover. And they are not the important part of DNA. The important part is what we find in the middle. It's the pages. So when we talk about genetic information, we're actually talking about recipes. So if you think of DNA as a cookbook, you know that DNA has genes. Well, all of your life you said, well, I've had the gene for brown hair, or I have the gene for blue eyes, but you probably didn't know what a gene was. Well, think of a gene as a recipe. We've all seen recipes before. And every recipe has 2 parts. There's the instructions part, there's the ingredient part. So you can think of a gene as having 2 parts. You'll see that they're called alleles. And you have to get part from your mom and part from your dad in order to make the complete recipe. And just like we use recipes to make things, genes are recipes. And they're recipes that tell our bodies how to make proteins. And that's what makes every single cell in nature different, is what kind of proteins it makes. Now in your body, you started off as 1 cell. Now 1 cell was called a zygote. And then you did cell division. You made copies of your DNA over and over and over and over and over again. But at some point in time, 1 cell said, you know what, I'm going to use the recipes that are in chapter 1. While another cell says, you know what, I'm going to use the recipes that are in chapter 20. Well, they're 2 different chapters, so they have different recipes, so they make different proteins. And that's what makes your cells different. So your brain cells use different genes than your stomach cells do. So your brain cells make this kind of protein, and that's what makes them brain cells, and your stomach cells make a different kind of protein, and that's what makes them stomach cells. But DNA is a cookbook that holds your recipes, and those recipes are genes. And genes tell you what kind of protein that you're going to make. So DNA determines what traits you have by determining your proteins. And as humans, some of our proteins are exactly the same. So, some of our recipes are exactly the same. You could go to a bookstore and buy 20 different recipe books, and they probably all have 1 recipe that's exactly the same. Might be called something a little bit different, but if you read it, it's the exact same recipe. Well, we're that way. That's why we have the same bones. That's why we have the same heart. But some of our recipes are a little bit different. Like one cookbook might have a chocolate chip cookie recipe that uses bittersweet chocolate, while another cookbook has a chocolate chip recipe that uses semi-sweet, while another one uses white chocolate chips. They're still all chocolate chip cookies, but they're a little bit different. And that's what makes all of us different people. So when we talk about a gene is a recipe in a cookbook. It's a section of DNA that codes for 1 protein. So 1 gene equals 1 recipe. And remember, proteins are also called polypeptides, because proteins are amino acids that are held together with polypeptide bonds. So when we talk about DNA, think of DNA as a cookbook with about 80,000 pages in it. It's been theorized that our DNA has 80,000 genes. Now, we only use about 40 to 50,000 of them. The rest is what we call junk, and we believe that they are kind of leftover. So it might be genes that or recipes we once used way back when, But as evolution happened, we stopped using them. But nature doesn't want to quite get rid of them yet. Now, like I mentioned, remember the number 3. DNA has 3 letters in it, and DNA stands for deoxyribonucleic acid. This is found in the nucleus. Remember, the whole purpose of our nucleus is to act like a safe. We are eukaryotic cells, which means that our cells have a nucleus, and so our cells protect our DNA. And if you think of DNA as a cookbook, you have to put the cover together. You have to put the pages together. And so the monomers, remember the monomers are like one Lego that I use to build other things. DNA is a polymer. It would be the entire structure. And the bricks that I stack on top of each other to make DNA are called nucleotides. So remember, monomer, think of it as just one Lego. DNA is a polymer. It's got lots of Legos that we put together in order to make the structure. But when we look at a nucleotide, again, a nucleotide has 3 parts. So nucleotide has a phosphate, a 5-carbon sugar, which is deoxyribose. That's where DNA gets its name from. And you know that deoxyribose is a sugar because words that end in OSE are sugars. And they have something called a nitrogenous base. DNA has 3 elements that make it up. It has carbon, hydrogen, oxygen, that's the sugar, phosphate, and nitrogen. So in the case of the nitrogenous bases, these are what make up the pages. The sugars and phosphates, these are usually drawn as kind of like an L. So if you were to tilt this, or if you hold your fingers like an L, your index finger, your pointer finger will be the phosphate. The bend or the elbow by your thumb would be the sugar, and then your thumb would be the nitrogenous base. And so these Legos kind of click together. So going down the side of DNA, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. That's what makes up your book covers. If the phosphates and sugars have nothing to do with your genetic material. If they did, we'd all be the exact same person because it's just phosphate, sugar, phosphate, sugar, phosphate, sugar. That's why you don't judge a book by its cover. The cover is not the important part. The nitrogenous bases line up, and these are what make the pages of the cookbook. So these are the recipes, as you're going to see in a minute, that your body actually reads. So other than remembering the number 3, here's an exception to the rule. On DNA, you have 4 possible bases. You can have a chemical called adenine, which will be abbreviated as an A, guanine, which is G, cytosine, which is C, and thymine, which is T. Here's another picture of a nucleotide, like I said, if you form an L, phosphate, sugar, and nitrogen base. So when we make DNA, the phosphates and the sugars click together. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. And that's what forms your book covers. That's what's going to protect the pages on the inside. That's the purpose of a cover. The nitrogenous bases, these letters, A's, G's, T's, and C's, are what actually make the words. They're what make the recipes that we read. And it took people a little while to figure out, okay, what are these? How do we read them? But just like when you were little, when you first started to read, you recognized something was a straight line or a curve. And then one day you woke up and you're like, oh, that's an A. And then one day you learned to put letters together and all of a sudden you have words. Well, we know that when these nitrogen bases line up, they form a sentence. And our body reads that sentence, and that's the recipe, that's the gene that they use. Now, they are divided into 2 groups. You have what's called adenine and guanine, and then you have cytosine and thymine, purines and pyrimidines. And for years, people had an idea of how DNA was made. I mean, we could dissolve it, we could crystallize it. So we knew that there was the sugar, we knew that there was phosphates, and we knew that there were these nitrogen bases. But we didn't really know how they all came together until a gentleman named Chargaff was able to show us. Now, if this is DNA here, you have the circles are the phosphates and the pentagons are the sugars. And so you can see it goes sugar, phosphate, sugar, phosphate, sugar, phosphate down the sides. So those are your book covers. Okay? That is not your genetic material. But then you have all these letters down the middle. Those are your pages. So just like when you have a book closed, you have a left page and a right page. So you can kind of think of that as you have a recipe on the left side, you have a recipe on the right side. And those are the words, those are the letters that you're going to read. Well, something has to hold our book together. You know that the book cover is solid, the pages aren't flying all around. So when DNA gets made, the nucleotides combined by a process called condensation. Remember, anytime you see condensation, it's also called dehydration synthesis. It's also called condensation synthesis. This is how things get put together. And they're held together by covalent bonds. So the covalent bonds would be what's holding the sugars and phosphates together. It'd be like you clicking your Legos together. Well, something keeps our book closed. Okay? When you're done with the book, you close it. That way, the pages don't get messed up. Okay? This book is not going to open by itself. Okay? So that's going to protect those pages. And so something holds our DNA closed in order to protect those letters that we have on the inside so that our recipes don't get messed up. And that's these things that we call hydrogen bonds. Now, a gentleman named Chargaff was able to show us that certain things fit together like pieces of a puzzle. So base pairs will fit in what's called base pairing. They're also called complementary base pairs. It's also called Chargaff's rule. But what we were able to figure out is that thymine only clicks together with adenine, and guanine will only click together with cytosine. So this is what we mean when we say they're complementary. So as you can see in this picture, they're like pieces of a puzzle. C's and G's will fit together, and T's and A's will fit together. And that's what we mean when we say that it's complementary. So anytime you have a C on one side, you're going to have a G on the other side. If you have a T on one side, you're going to have an A on the other side. And again, this is what we call base pairing rules. A's always go with T's, C's always go with G's. And it's also what we call Chargaff's rule, named after the gentleman who was able to figure out the correlation between these nitrogen bases, as you can see here. Okay. Chargaff's rule, again, any DNA molecule must contain equal amounts of adenine and thymine and equal amounts of cytosine and guanine. So again, whatever you have on the left, its partner is going to be on the right. And they have to fit together like pieces of a puzzle. They have to be complementary to each other. So this is your genetic information. The sugars and phosphates going down the sides are your book covers. And they're just there to protect the pages on the inside. And the pages on the inside have the words that we read in order to make our recipes. And so as you can see here, these chemicals that we call nitrogenous bases are going to be what make up the words that our body uses to read the recipe. So if a strand is 15% guanine, how much adenine, cytosine, and thymine do you have? Well, obviously we have to have 100%. So if guanine is 15%, what always goes with guanine? Well, G's and C's always go together. So if I have 15 green Legos, its partner is the cyan Lego. So if I have 15 greens, I have to have 15 C's as well. Well, 15 plus 15 is 30, which means I have 70% of my DNA are A's and T's, and you just divide them in two, and that's how you can figure out the rest of your percentage. But wherever you have G's, you will also have C's, and wherever you have A's, you will have T's. Now, when we talk about mutations, a mutation by definition is a change in DNA. And when people talk about mutations, it's these words, these letters that get messed up. And we're going to talk more about mutations in a moment. Now, like we mentioned, the DNA structure is always called a double helix. Double meaning a twist, or excuse me, helix meaning a twist, and double referring to the two sides. So what makes up the sides of the ladder, or what I call the book covers, are your sugars and phosphates. What makes up what people call the rungs of the ladders, which are what you hold and step on when you climb up a ladder, is what I call the pages of the cookbook. Those are going to be made of the cytosine, the guanine, the adenine, and the thymine. Those are your nitrogen bases. So that is your genetic material. That's going to be the part of the DNA that you read. Now, the double helix structure, again, for many years we had an idea of what DNA was made from, because we could dissolve it, we could crystallize it, we could analyze it. And we knew it had phosphate. We knew it had this sugar, and we knew it had these chemicals, but they didn't really know how they came together. And Rosalind Franklin was an X-ray chromatographer. She would take X-rays of crystals that formed from different chemicals. And Watson and Crick, the two gentlemen who were given credit for discovering the structure of DNA, used her work, her X-rays, in order to figure this out. Rosalind Franklin died from ovarian cancer as a young lady, probably due to the X-rays that she was taking. But Watson and Crick were able to show us that what they called the sides of the ladder Again, that's what I call the cookbook. That's what I call the covers of the cookbook. It's sugar, phosphate, sugar, phosphate, sugar, phosphate. And they're held together by these things called covalent bonds. So everybody in nature has, or everything in nature has the same sugar, phosphate, sugar, phosphate. So the covers are not what make us different. We all have the same book covers. It's the pages on the inside. It's the recipes that we have. So humans have different recipes than dogs have, which have different recipes than plants have, which have different recipes than a mushroom has. It's the ladder rungs, or what I call the pages of the cookbook, that makes everybody's DNA different. And these are your nitrogen base pairs, and these are what contain your hydrogen bonds to hold them together. So as you can see here, bases bonding in the center of the helix hold the double helix together. Hydrogen bonds are always drawn as dotted lines. And you can see how the nitrogen bases are often drawn as puzzle pieces. So adenine and guanine, guanine and cytosine will match up, and adenine and thymine will match up. And you can see sugar, phosphate going down the side. Okay. The nitrogen base is attached to the sugars, and it does have these book covers, and it has these twists. Well, the hydrogen bonds are there to protect the pages. We want to keep these pages protected, so we want to keep the book shut. I remember growing up, my grandmother had a Bible that actually zipped so that the book wouldn't open up by itself because these pages were so delicate. Well, think of this cookbook like this. It's very, very, very delicate. So we use these hydrogen bonds to make sure that our book doesn't fly open and damage the pages on the inside, because that's what happens when you have a mutation. Those pages get damaged. So again, the sugars and phosphates make up the sides of the ladder. So those are the book covers in order to protect the nitrogen bases. The nitrogen bases are the pages in the book. These are your recipes. They contain the important information and the material. So when we talk about genes, genes are recipes that tell us how to make proteins. So your genes are made from nitrogen bases. And so again, we're remembering the number 3. When we look at DNA, we've been able to figure out what the recipe says. So it reads like a sentence. When you have a chain of nitrogen bases, as I have here, AAA, CCT, GCA, TCA, our bodies take this chain of letters and divides them into groups of 3. And what we find is every 3 nitrogen bases is what we call a triplet. So AAA would be a triplet. CCT is a triplet. GCA is a triplet. TCA is a triplet. And what triplets tell us is one ingredient. Remember, proteins are made from amino acids. Well, this gene is a recipe, and this recipe tells us what ingredient we need in order to make that protein. So when your body sees a gene, it takes that gene and divides it into groups of 3s. And every 3 letters is called a triplet, and that tells me what ingredient I need. So if this were a gene, this would tell me that this recipe needs 4 different ingredients. So when you're studying DNA, just remember the number 3. DNA is deoxyribonucleic acid. It is our genetic material. It's a cookbook that holds our genes. And genes are the recipes that tell us how to make proteins. They're made up of nucleotides. There's 3 parts to a nucleotide: a sugar, a phosphate, and a nitrogen base. And the nitrogen bases are the words, form the words that our body reads in order to make the protein. So we divide that sentence up into groups of 3s, and that tells us what ingredients I need.